this was a collaborative effort between the Regev Lab and Jesse Boehm's group, and me and JT Neal sort of led this study. And what we wanted to understand was what different cancer mutations do, because millions of them have been sequenced across different patients. And it's really key to understand which mutations actually are important for the tumor growth and will affect specific treatments assigned to patients, and which ones are sort of more bystander mutations that are not actually affecting the tumor. And this important question has been experimentally tackled in many ways, traditionally through specialized functional assays that determine for a given gene whether mutations in it change the functionality of that gene, maybe making it a loss of function or a gain of function. This is hard to do because such an assay needs to be developed for each new gene of study. And on the other hand, there are computational efforts to try to predict the effects of mutations across a variety of genes. But at the end of the day, these have to be tested experimentally. And to do that, we needed to move away from sort of readouts that measure only one specific function and find something more general. And what we settled on was gene expression as a readout of mutation impact. So what we do is we take cells and we overexpress a specific gene, either the wild type or a specific variant in that gene. And then we look at the expression profile of the cell. And essentially, we can use the cell as a biosensor of variant impact, because if overexpressing the variant leads to the same types of expression profiles as when we overexpress the wild type version of the same gene, then it means that the variant didn't really affect the gene. So it means that it didn't really have any impact. Whereas if the gene expression profile in the cell changes dramatically, it means that that variant actually changed the normal function of the gene. Maybe it led to a loss of function, or maybe it just created a whole different function. And the way we scale this up was using single cell screening technologies, in particular Perturb-seq, which allow us to do this experiment and profile hundreds of variants in one single experiment. So essentially what we do is that we do the experiments I talked about, but in a pool where each variant has a little barcode attached at the end. Then each cell gets to overexpress one variant, and then we do single cell RNA sequencing to get for each cell not only its expression profile, but also the identity of the variant that is overexpressed in that cell. So then all we have to do to know if a variant has an impact or not is basically look at the gene expression profiles of cells that overexpress that variant and compare that profile with cells that overexpress the normal gene. So we have used this approach to study 200 variants in two quite different cancer genes, PIF53 and KRAS. And in PIF53, we were able to identify many of the loss of function variants, whereas in KRAS, we recapitulated gain of function variants and also learned more about the specific pathways that these variants affect. In summary, we provided sort of a gene agnostic method for studying coding variants, and we applied it to cancer, but it should be applicable sort of more generally to study gene function. And we're really excited to basically deploy this further to understand the vast number of coding variants in cancer. When we started this project, we were expecting to find variants that either had a very strong effect or they had no impact and they looked exactly like the wild type version of the gene. However, what we actually found was that we found variants all the way in between these two categories. For example, for KRAS, I'm showing you here all the variants ranked by their effect sizes. At the top are the variants with the lowest effects, and on the bottom, the ones with the largest effects. And if we look at, for example, a gain of function signature score, which we call here PC3 for principal component 3, and we can compute these scores across each single cell, and then look at how the distribution of these scores changes for each variant in red compared to the wild type in gray. And as I mentioned, we see that the variants that have no effect have this distribution exactly the same as the wild type one, whereas the ones with the strongest effects completely separate themselves. They have much higher gain of function scores compared to the wild type. The interesting thing was that there was everything in between. So there was really a gradation of the amount of gain of function that a variant can exert. So this allows us to sort of add nuance to the idea of either driver mutations or passenger mutations, and really think about variants as having more of a continuous type of effect. The next very exciting thing is to study variants using base editing at their endogenous locus. And there's many reasons why that's exciting, and there have already been some studies going in this direction. So I think the reason why it's interesting is because then you could look at multiple variants in different genes and how they interact with each other in the cancer cell. And we can also start to look at the effects of variants, not only on individual cells, but more on the tumor microenvironment.